The COVID pandemic has affected the lives of everyone to make 2020 a year unlike any other in our lifetimes. Governments have had to balance the freedoms and functions of society with protecting the population from an out-of-control pandemic. The public try to follow the news to see where things are heading for them in the coming weeks, but it's hard to understand variants, mutations, PCR tests, and so on without an education in molecular biology. This video will try and explain these issues in simple terms so that you can make more sense of the media reports and the changing restrictions to our daily lives. This video will cover 4 areas of science so you'll be able to follow the facts behind the global pandemic. Firstly, I'll describe the main features of the SARS-CoV-2 virus which causes the COVID-19 pandemic. I will then explain the PCR test that's used for COVID-19. I'll explain how it works and how unexpected results in these tests are linked to the rapid spread of a new variant of COVID-19 in the United Kingdom. I'll then go on to explain how mutations arise and how they affect the behaviour of the virus. Finally, I'll describe 2 mutations found in rapidly spreading coronavirus variants that are causing concern and a new wave of strict lockdown restrictions. You can use the timestamps in the description below this video to jump to any section to watch it again. Feel free to ask us any questions in the comments section below. What does the SARS-CoV-2 virus look like? Well, it's a coronavirus. There are 4 other human coronaviruses that cause the common cold and 2 other newer coronaviruses that cause MERS and SARS. The coronavirus is very small. A human hair is approximately 750 times thicker, and it looks like a ball covered in spikes. So it's spherical, and the sphere is a membrane of a fatty lipid substance similar to the surface of our cells, and embedded in that membrane are different proteins, including the spike protein that sticks out to form these spikes, and these are really important for the virus's ability to interact with human cells. And then inside this envelope, what it's actually delivering is the genetic material that encodes the virus. So this is the rogue code that tells human cells to make virus. And this is an RNA genome, and I'll get into explaining what RNA versus DNA is later on. The spike protein mediates the binding of the virus to human cells via a protein found on the surface of some of our cells, and this protein is called ACE2. So the virus sticks to this, it enables it to engage with human cells, and then it will go on to progress to invading human cells. ACE2 is a really important protein to us. It's actually an enzyme that reduces blood pressure, and it's attached to the outside of cells in these tissues. So for example, it's found in the lungs, it's also found in the heart, it's also found in our blood vessels and many other tissues besides. And where it's found also aligns with the kind of illnesses that you can get if you're unfortunate enough to have COVID-19 and the illness progresses beyond fever. So you could have respiratory failure, various forms of heart failure, and also you can have vascular defects and failures as well. So it's an illness that can cause much more than just a cough or a change in sense of smell or a fever. It can go on to do an awful lot of damage and lead to death. So what does the life cycle of the coronavirus look like? Now I've simplified this, but the main steps are that firstly it binds to this ACE2 receptor found on the surface of our cells, such as in the lung. It then the whole particle makes its way into the cell, And when it does so, it delivers this rogue code, this viral RNA genome. And this genome encodes a small number of proteins, and these proteins make up the virus itself, so the sort of physical structure of the virus. They make enzymes that will replicate the viral RNA. And also there's a bunch of proteins in there that serve to hijack host cell processes. So tell the cell not to spend time doing what it would normally do, and please divert all of its energy and attention to making virus. Thank you very much. And that's a large part of why our cells get so unwell. So the virus makes these proteins. It then goes on to make copies of its genome, and then it will assemble its genome into new virus particles, which are called virions. And then these mature, and then they'll eventually bud out of the cell and leave the cell. And our human cells therefore become factories for making virus. It's important to note that the virus isn't a living thing. It's not capable of doing anything on its own. It's just an oddity of nature, in that this rogue code can make this particle, and this can go round and round. What does the genome look like? So this is the RNA code of this virus. It's very small. It consists of less than 30,000 letters, or we call bases, of genetic code. And it contains the absolute minimum required to create a virus. So it only makes about 29 proteins versus over 20,000 proteins that would make a human being or any other complex animal. So for example, the human genome is over 100,000 times bigger. So this is a very small genome. It's very compact. It squeezes in a lot of proteins into a very small amount of space. And the kinds of things it makes are the physical proteins that are found on the viral particles themselves. For example, the spike protein. It makes a bunch of enzymes that serve to make the copies of the viral RNA. And then there's a whole bunch of proteins here that are involved in the life cycle of the virus, and many things in here that hijack human cell processes. So, very clever, very well organized, neat and tidy genome. So, how do we detect the SARS-CoV-2 virus in people when we suspect they've got COVID-19? So, people will present with symptoms, typically a persistent dry cough, a high fever, difficulty breathing. In some people, they only get a loss of sense of smell and taste. If you present with these symptoms, you'll report to your local healthcare provider, book yourself in for a test. And typically test you'll get given is a PCR test. And this test is looking for the viral RNA, that viral genome, present in a patient's airways. And typically you will have a long cotton bud and you will wipe it at the back of your nose, which is very unpleasant but important. There's plenty of virus tends to sit at the back of the nose. And then this gets put in a tube, and it can stay in the fridge for 2 or 3 days, no problem. And then that has to go to a laboratory for this test to be carried out. And in the laboratory, the RNA is chemically extracted from the sample, so it's removed, so that the virus is destroyed and any other human cells that are in there get destroyed. And you just end up with RNA in a tube. This RNA is converted in the lab through a chemical process to make a DNA copy. Again, I'll explain DNA and RNA in a moment. So we make a DNA copy of the virus genome. And once we have this DNA copy, we can then subject it to an assay called PCR, which I'll explain in a moment. Now, there are other tests for COVID-19. There are what are called serological tests. So these are blood tests, and they test for the presence of the virus or antibodies, our antibodies against the virus, and looking for either of those in our blood. And then there are some rapid tests called lateral flow tests, and these look like pregnancy tests or drug wipe tests, depending on what you might have seen before. So these are pieces of paper with different chemicals on them, and they're trying to detect viral RNA or antibodies typically. So these are really useful as a rapid test. They're at the point of care. You don't have to go to a laboratory to do it, but typically they're nowhere near as sensitive as these laboratory tests. So I'm just going to focus on this PCR test in this presentation. So a PCR test needs to be performed early in the early stages of a COVID-19 infection. So typically we're calling here, this line here, the point where symptoms have presented and someone's booked themselves in for a test. They may have been exposed to COVID-19, to the virus 1 or 2 weeks earlier, and then this virus has took time to infect us and multiply and grow inside us. And as time goes on, you'll get an increase in viral particles in your airways. And so the amount of viral RNA in pink here accumulates most around the time of when symptoms present themselves. So the days immediately before and after when symptoms present themselves. If you take a swab from the nose, then the viral RNA does last for longer in that part of the body. If you're testing for antibodies, these only accumulate in the weeks after an infection. So testing for antibodies is an excellent way of seeing whether somebody had COVID-19 infection in the past, but is not a good way of seeing whether somebody has got COVID-19 right now and presents a risk to society. So how does the PCR test work? Well, I'm assuming that a lot of people watching this video do not have a higher education in molecular biology. So I'm going to try to explain it to you in simple terms. And to understand PCR, we need to know a bit about DNA first. So DNA is the genetic material, the code inside all cellular organisms, whether it's a bacterium, a plant, an animal, a human being. We all have DNA as our genetic material. DNA is packaged into chromosomes, you may have heard of, and then when these chromosomes are unwound, what you find is that DNA is this highly characteristic structure of a double helix. It's two strands wrapped around each other. And within these strands, and the strands what we would call a sugar phosphate backbone, so that's the strand itself. Inside those strands are bases, or we can just call them letters in terms of a language. These are the genetic code, these molecules. And there are 4 bases. There's adenine, A, thymine, T, guanine, G, or cytosine, C. So we're just going to call these the letters A, T, G, and C. And that's all the genetic code is. There is only 4 letters. And there's a strict rule on how those letters can interact with each other. So a letter from one strand physically interacts with a letter on another strand. And only A and T can bond with each other, can interact with each other. And only G and C can interact with each other. So these have 2 bonds, these have 3 bonds. So A cannot pair with G or C, T cannot pair with G or C, and vice versa. That's a strict rule, and it's true for all organisms. So you can see here, here's our code on one strand. T pairs with an A pairs with a T, C pairs with a G, G pairs with a C, and so on. Quite straightforward. The beauty of DNA is that we have 2 copies of the code. We've got 2 strands of it. So that's very safe. If one gets damaged, we've got another one with which to read when repairing a problem in DNA. And these 2 strands are held together by hydrogen bonds. Hydrogen bonds are not very strong bonds, but collectively, because there are so many of them between all these bases, it forms a strong link between these 2 strands. DNA is very stable. And you can imagine it a bit like Velcro. You've got 2 straps of Velcro. When you stick 2 long straps of Velcro together, that's very, very firm. It's very strong. You can carry quite a lot on Velcro. But if you peel them apart in just the right way, each of those bonds, each of those sticky bits can pull apart quite easily. And so DNA is like that. It's very strong, but quite easy to pull apart when you know how. So what's the difference between DNA and RNA? Well, DNA is double-stranded, as I've just said. RNA is a very similar molecule that's only single-stranded, typically. And the only difference between DNA and RNA is that one of the bases, a different molecule is used. Uracil is used instead of thymine. It looks very much the same, and the base pairing rules are still the same. So instead of having a T, we have a U. And in the case of RNA, U pairs with A. T and A pairs with U. Okay, so T and U have been swapped with each other. So why would a coronavirus prefer to use RNA for its genetic material rather than DNA, like all cellular organisms? Well, DNA is really stable. There are 2 copies of genetic information, which allows repair. If we lost a base here, we could copy from the other one. And a problem for the virus is that it's also a heavier, stiffer molecule than RNA. You can imagine DNA a little bit like a rope tying a ship to a harbour. It's very long, very, very strong, but it's quite hard to move. It's quite hard to bend. It's very thick. Whereas RNA is very, very flexible, like a flexible cable. And so it's much lighter, much easier to package inside a small virus. Remember, a coronavirus needs to float in the air. It's mostly transmitted through the air. So it needs to be a light virus, a very small genome that fits into a small space. Another advantage of RNA for the coronavirus's point of view is this is far more prone to mutation. If there's a loss of any information here, then it's gone and the virus has changed. And this is exactly what the virus wants to do. It's in a constant war with our immune systems, and our immune systems will eventually get smart enough to recognize the current COVID virus that we're being exposed to. Well, the virus will want to change. If it can't change, it doesn't exist. So, how does PCR work? So PCR stands for polymerase chain reaction. And essentially, it's just a way of making lots of copy of a known DNA sequence. So for example, you want to see whether there's a gene for the coronavirus spike protein. Say you're interested in that. PCR allows you to make lots and lots of it in a tube. And there are several things you need. Firstly, you need to start with a DNA template. You need at least one copy of something that you're going to expand upon. So it's like you've got a copy of a book and now you want to make millions of copies of that book. So in this case, our DNA copy of the viral genome in the test sample is our DNA template. Next, you need knowledge of what sequence you want to amplify. Well, we do know what the sequence of the coronavirus genome is. So if we want to amplify the spike protein gene, we know what the sequence of that is. And that informs us how to design these short pieces of DNA that are going to be single-stranded. And we make these in a laboratory. And these are called primers, and you'll see them in the next diagram. And these contain sequences that match what we want to amplify. So we'll have one primer that matches the beginning of the spike protein gene, for example, and another primer that matches at the end. And we are going to amplify the sequence between those 2 primers. Then we need the building blocks of DNA. So we need those 4 bases, A, T, G, and C. And then we need something going to do the work for us. And this is a protein called a DNA polymerase. And this is an enzyme, and it's essentially the protein that's going to build the DNA chain for us. And importantly, we use a DNA polymerase that only works at high temperatures, specifically at 72 degrees Celsius. So our bodies are work at 37 degrees, and our enzymes are optimized to work at 37. And at 72, they would be dead. They wouldn't work. So we use enzymes that come from bacteria that grow in the hot waters that come out of volcanic vents, amazingly. And so that's where these DNA polymerases come from, and they like to work in hot temperatures. And you'll see why that's important in this diagram. So this is a cartoon here showing those things I've just mentioned. So in a PCR, you just have one tube and you're sticking it onto a machine which has a metal block in it that fits that tube. And what the machine does is capable of heating and cooling that tube to very specific temperatures very, very quickly. And that's all that machine does. And in the tube, you've got this cocktail of your starting template. So this is our DNA copy of the viral genome. We've got these short DNA primers, which match the target we want to amplify, in this case, perhaps the spike gene. We've got the individual bases, which are shown as purple T's here, but they represent the A, T, G, and C bases. And then we have the enzyme that's going to do the work for us. So what happens in a PCR? Well, first thing is we heat the whole cocktail up to 95 degrees. This enzyme is still okay in that temperature. But what happens is the amount of heat there, it's capable of melting the hydrogen bonds between the two DNA strands. So the strands themselves stay intact, but they become separated from one another. They just peel apart and float away from one another. And this is called denaturing. We melt the strands. So that exposes the genetic information that's on the inside. So the bases are now exposed, ready to stick to something else. And if we just cooled this down, then these two strands could go back and stick to each other again. But because we've got these primers here, if we just cool down not to room temperature, not to 20 degrees, but cool it down to 60 degrees, then that's still hot enough to keep these two strands apart, but cool enough to allow these primers to interact with the matching sequence. So if the spike gene is present in our starting template, so somebody is COVID positive, then this primer will interact. If somebody isn't infected with COVID-19 and the viral RNA isn't present, this primer will have nothing to stick to and nothing is going to happen in this reaction. So this cartoon shows that we do indeed have the viral DNA sequence present here. So the primer sticks. The next stage, so this is called annealing. The next stage now is when the polymerase does its job. So we increase to 72 degrees now, and suddenly now the polymerase is able to work. And what the polymerase looks for is this kind of scenario where you've got a double-stranded DNA template. So you've got the two strands, but there's a gap. There's an area to fill in. And the polymerase will interact with this, take all these individual bases and add them to their matching partners. So A will go with T, T with A, G with C, C with G, and it will add these on all in one direction, and it will complete the product. And when we've done that, what we end up with are now two copies of the DNA target we wanted to amplify. So we started with one, now we've got two. And if we just repeat this, these temperatures go 95, 60, 72, and go back round again and round and round again. As we repeat these cycles, as we call it, we accumulate, we double the amount of product every cycle. So 2 becomes 4, 4 becomes 8, 8 becomes 16, and so on. And we get this chain reaction of amplification of the target we were intending to get. And this enables us to see whether the viral sequence was present or not. So how are we going to do that visualization? The problem with all these molecular biology techniques is that the molecules are incredibly small, we don't have very much of them, and generally we're just working in a small bead of colorless liquid. We actually can't see anything. So we always have to design something in, a kind of reporter that tells us what's going on. We need some molecule in there to say, hey, you've got what you wanted. So we would be able to see that we've made some DNA in the tube. The problem is we don't know which DNA we've made. So one way we can figure out whether that viral spike protein gene was being amplified in PCR or not would be to add a third short DNA sequence, which we call a probe. So it's another piece of DNA that binds to, for example, the middle of the spike gene. And if we just go down here, so we've got one primer that binds to the beginning of the spike gene, one that binds to the end. Now we're adding a third one in the middle. And the key concept here is that this middle probe actually has a reporter on it. It has a chemical on it that's fluorescent. And fluorescence is a kind of luminescence, it makes light. But specifically what fluorescence is we shine one colour of light at a tube, and if this molecule is present and is fluorescent, then it will emit a different colour of light. So for example, we might shine blue at the tube and green would come out. And in this case, we've got a molecule on here that will make green fluorescence, for example. But at the other end of the probe is another molecule that absorbs that green light, and it's a quencher. So the probe on its own is dark, we can't see it. But if for any reason these two molecules became separated from each other, then we will be able to see the fluorescence coming from this molecule. And this works in our PCR assays because you will see in the PCR, the polymerase would normally bind to this primer and extend it all the way to the end and create and fill in and create the full double-stranded DNA molecule. But this probe is now in the way. It's bound and it's in the way, and it might be a spanner in the works. But these polymerases are not stupid. They've evolved to make sure that no little bits of DNA that might be bound in the way would actually interfere with what they are doing. And the polymerases are able to chew up these pieces of DNA. So if the polymerase comes along here and sees this, it will cut this up into the individual bases and spit them out, and then complete the whole sequence. And that's indeed what it does. So on this strand, the polymerase is bound to a primer and is able to go right to the end. On this strand, it's bound to the primer, it's carrying along, it sees the probe, chews it up, spits it out, and then completes the product. As it does so, it releases one molecule of that fluorescent reporter. So in other words, every time we do the PCR once, we get one molecule of fluorescence. So as the PCR goes on and we make more and more DNA copies, we release more and more fluorescent reporter. And we can see this in our PCR machine, because our PCR machine shines light and reads the color of light on our PCR action tubes after every single cycle. And we can see this accumulation of green fluorescence. So if we get this fluorescence, it means we've got this spike gene in the tube. And the amount of fluorescence we get is directly proportional to how much of that viral sequence was there in the beginning. So if we get more of it or less of it, we can say the person had more or less viral RNA. Okay. So this here is the test, the most commonly used test being used in clinical laboratories in the United Kingdom. And it's called the TaqPath COVID-19 RT-PCR test. And it's made by a company called Thermo. And what Thermo did was to design sets of primers and probes that pick up 3 different genes shown in these little orange bars across the COVID genome. And the idea would be that you want to get all 3 to come up positive. So if you get a 3 out of 3 come up positive, that person's most definitely positive. And these assays all have a different colour of fluorescent reporter. So we've got one that'll be green, one that'll be yellow, one that'll be red. And so in the PCR reaction, the machine will show these curves of red, green and yellow, and they all come up and they're all positive. So this is a positive test. The number of PCR cycles it takes to reach a threshold. So a line is set that says if you cross this line, it means the PCR's worked. it means you're positive for that target. So as soon as you cross this line, you're positive. So the number of cycles, so if we draw a line down here to get to this point, this is called the CT, the cycle threshold. The more virus that's present in the patient's sample, the fewer cycles that would be required to reach this point. So the more DNA we've got here, the quicker the PCR is going to get to that point. If there's less DNA, it's just going to take a bit longer. The PCR will still be positive, but it will take longer. So lower CT values mean more virus. So that's quite important later on for understanding some data that's been coming out of the UK labs about some of these new variants of the coronavirus. And I'll talk about that in another talk. But that's why I'm referring to CT values here. Okay, let's move on. So, mutations of the coronavirus. They've been found all across the genome. So this here is a plot from taken from all of the genomic sequencing of coronavirus samples all across the world. And they've all been submitted to databases, and this is data collated together in this database called GIS-AID. And we can see that mutations are found all across the coronavirus genome. And this is the frequency of them here. So almost every point of it can get mutated. Are all of these dangerous? Are only some of them dangerous? How can we tell which of these mutations are worrying? Is just having a mutation worrying? Or are there some that we should focus on? So, to understand this, we really need to know how DNA codes for proteins. We need to understand the nature of mutations. So I'm going to take you through this fundamental biology. So now we know about DNA and we know about RNA. So this slide shows you what's called the Central Dogma of Biology. This is how the genetic information encoded in our genes is converted and turned into functional proteins in our cells. The DNA genome is just a set of instructions. It's a library somewhere. But the functions of our cells, of our body, come from the proteins. And the way it's done is it happens in two stages. Firstly, we make a copy of the DNA code for any given protein we want to make. And this copy is called a messenger RNA. The reason we make this copy is the DNA code is really extremely valuable. And we don't want to just keep throwing this around the cell as we want to make copies of several hundreds or thousands of proteins. So this is our archive we take care of. This mRNA is like a little chitty, a little instruction, an order, a purchase order, as it were, to say, please make this protein. And this is sent off to a different part of the cell where the protein factory is. And if you look at the sequence of this mRNA here, it matches the DNA. So this DNA gene, this is its code. It says ATG, AGT, and so on. And the mRNA says the same thing, it's just that the T's are now used. This mRNA needs to get turned into a protein. And the way that that's done is through codons. So sets of 3 letters at a time, 3 bases, code for 1 amino acid. Proteins are made, the building blocks of proteins are amino acids. So the key thing are these codons and these amino acids. So there are 20 amino acids that are the building blocks of all proteins, which is quite an amazing thing to think. All of nature, everything you know, everything in the natural world just comes from these 20 amino acids. So you could make strong proteins like your nails, the hooves of a horse. You could make flexible, long proteins like your hair, or you could make colourless proteins like the lens in your eye, or you can make a protein that can bind oxygen in your blood. It's an amazing number of things that can be achieved from just this toolkit of 20 amino acids. And these all have different properties of charge, ability to interact, shapes that they can form. Some are small, some are large, some are twisty, some are not. So you have this chemical toolkit that can make proteins. And this assembly of proteins happens in a sequential manner. You start at the beginning and you add on one amino acid at a time, and you make a long chain. And that chain of amino acids then folds up on itself and makes a structure that performs the role of that protein. And there are special molecules that can recognize these 3 base codons in RNA, and they then bring in each one of those amino acids. So there'll be a molecule that recognizes AUG and it would bring in methionine. Another one that would recognize AGU, bind to that and bring in serine and deliver that to the protein machine, building machine. So this is the genetic code, and the genetic code is the same for all species, whether you're a bacterium, plant, animal, human. So here are the same 20 amino acids, and we can abbreviate their names as either a single letter or as a 3-letter code, but it's the same 20 amino acids. And here, these are the 3-letter codons that encode for each of those amino acids. So let's just think about this code and compare it to a language like our written language. The DNA RNA bases are the letters. There's only 4 letters. And the codons, the set of 3 letters, are the words. So all the words are only 3 letters long. So there are 64 possible combinations of making a codon from 4 bases. So in other words, there's 64 words in the vocabulary of the genetic code. But there's only 20 amino acid outcomes that are possible. So the code is degenerate. So you'll see that some amino acids have multiple codons that can encode for them. So alanine's got 4, argine's got 6. Some of them just have 2, others just have 1 codon. But in general, there are many codons that can lead to the same amino acid. And that's the genetic code. There are 3 codons that code for stop. So to tell the protein builder machine, okay, you've come to the end of the gene. This is the end of the protein. You can stop building now. And one of the codons is found at the start of every single protein, and that's the same one as methionine. So that's the start codon. So the protein builder machine gets hold of an RNA, looks for AUG, starts there, and starts building from there. So we're going to do some of that building now. So I just want to take you through this one example here, and then we'll mutate this and see what happens to the protein that we make. So in this example, I'm just showing you the first 4 codons, which make the first 4 amino acids of this made-up protein. So let's have a look at the code. AUG. Well, AUG is here, and that codes for methionine. So methionine is the first amino acid. Codon 2, AGU. AGU's all the way over here, and that codes for serine. So serine is the second amino acid. CCA codes for proline. That's the third amino acid. And AGU codes for serine again. It's the same one as number 2. Okay. So let's see what happens to this protein sequence if we change just one letter in the code. And I'm going to show you 3 scenarios that look really similar in terms of a mutation, but have 3 wildly different outcomes. So firstly, if we insert just one base at the start of codon 2 here, we're just going to sneak an extra base in. It's just one little base. The rest of the gene's the same. Can't make a big difference, right? So we're just going to stick an A in here. There it goes. So we've stuck an A in there. The rest of the sequence is the same. Right, let's read the code now. So AUG to start is methionine again. Fine. The next one is AAG. Right, we were serine before. Ah, right, it's not, that doesn't work anymore. So what's going on? Well, AAG is found here, and that codes for lysine. The next one, UCC, codes for serine. And then the next one is AAG, that codes for lysine. So if we can just go back, we started with methionine, serine, proline, serine, and we've ended up with lysine, serine, lysine. So even though we've just inserted one base, we've completely changed the protein outcome of this gene. Now, most proteins are hundreds of amino acids long. In this scenario, all of the amino acids will be different. And we're just making a junk protein. It's just gobbledygook. It doesn't code for any real protein anymore. It's just a piece of junk. So a one-letter change can have a huge effect on an organism, completely change a protein. In this case, we will lose the function of that gene. Okay, let's try a different scenario. Let's just swap one letter this time. We're going to swap the letter at the end of codon 2. So here's codon number 2, AGU, and we're going to swap this to an A. So it's now AGA. So AGU was serine. AGA, it's not serine anymore. It actually turns out to be arginine. AGA puts an arginine in. Now the other codons stay the same. So we've still got CCA, AGU, so we've still got proline, serine. The rest of the protein will be the same. So in this situation, a one-letter change in the code changes just one amino acid. And this is the most common kind of mutation you will see in any organism and see in viruses as well. And in this case, we've swapped a serine for an arginine. These are two very different kinds of amino acids. So in this case, we might have considerably changed the function of this protein. It might now be a different shape or have different interactions with other proteins. So it might do a completely different thing. So this mutation might be quite severe. Okay, but it's just one amino acid is changed. Okay, finally, let's just swap one letter now at the end of codon 3. So we're doing the same thing as before, but we're now at the end of codon 3, and we're going to swap this A for a G. So it was CCA, that's proline, but now we're moving to CCG, and CCG is still proline. So in this case, a one-letter change in the code caused no mutation at all. It was silent. And that's because of this degenerate code. If you swap one of the bases where there are multiple options of codons, and you swap just that last base of a codon, then you might have a silent mutation. So this hopefully is explained to you. It's quite complicated as you go through these things. Nature can be very complicated, but it explains to you why some mutations matter more than others. So the fact that we've got all these many mutations have appeared across the coronavirus genome doesn't mean we've got millions of different kinds of viruses, or there's going to be millions of kinds of different diseases as an outcome of those viruses. It turns out the vast majority of coronavirus mutations don't affect COVID-19 disease in any significant way. And that's because many mutations are silent, they don't actually change any proteins. Or if they do change the proteins, those changes themselves aren't that important, and the proteins still do the same job they originally did. Now, these silent mutations are still really helpful to us, because they allow scientists to track the evolution and spread of the virus around the globe. So let's say a new mutation appears in Madrid in Spain, we can see it spread to the other parts of Spain, other parts of Europe, and so on, and see the interactions between the virus and human behaviour. Now, some mutations in here might improve viral transmission or viral performance, so we must remain very vigilant. So what we have to do is see where those mutations are. Do they change the sequence of a protein? And is that protein likely to cause a problem when it has this mutation? And history has told us with viruses that these spike proteins are the ones to be most worried about. These are the proteins that are on the outside of the virus. These are the proteins mostly recognized by our immune system. And these are the proteins that mediate the initial engagement with our cells. So mutations that change the surface of the coronavirus spike protein might remove epitopes. These are binding sites that can be recognized by our antibodies. So this here is a molecular structure of the spike protein that's been solved. Mutations that are at the ACE2 binding site of the spike protein might improve the ability of the virus to stick to our cells and therefore infect us better. So these are things to worry about. And indeed, there are two spike, so there are lots and lots of spike mutations have been identified, the vast majority of which are quite mild and nothing to worry about. But two have appeared recently in the United Kingdom and in South Africa that are associated with a very quick spreading variant of the virus. So one of those is the N501Y mutation. So this is position 501 within the spike protein, and it was an asparagine, and the abbreviation for that is N, and now it's been turned to a tryptophan amino acid, which is abbreviated as a Y. So that's what N501Y means. We've swapped one amino acid for another. The rest of the protein is still the same. It turns out position 501 here is on the ACE2 binding surface of the spike. So this is a space-filling model of the spike, and here is a wire diagram model where you can just look at the inside of the protein. And in green here is the ACE2 molecule, and you can see how the spike protein changes shape and reaches up to bind ACE2. And this position 501 is right at the interface between our ACE2 protein and the virus's spike protein. So this mutation has arisen several times independently across the globe and is often accompanied with other mutations. But what's most concerning is that it's found in both of the fast-spreading new variants that have appeared in the UK. That one's called B117, and South Africa, and that one's called B1351, and these have appeared in the last few months of 2020 and have spread through those two countries very quickly indeed. And scientists currently think this mutation may increase binding to ACE2, but a lot of research will need to be done. This pandemic may be over by the time we understand how these mutations work, unfortunately. A lot of work needs to be done. Another mutation, an interesting one, involves the deletion of 6 bases that takes away 2 amino acids that are in positions 69 and 70 in the spike protein. And these are found here on the outside of the spike protein. Now again, these have arisen multiple times around the world independently and found with other mutations. But one of the times it's arisen is in that same B117 variant in the UK, the very fast-spreading variant. And another is in the mink farming community in Denmark. And again, that strain has spread across Denmark and neighbouring countries quite quickly as well. It's important to point out that this deletion is not found in the new South Africa variant that people are worried about. So scientists think that this mutation might alter spike recognition by antibodies, and this might impact some antibody therapies, but it's not clear why this deletion might lead to faster spread of the virus in itself. But the reason that I bring this one up is that it's actually quite intriguing, because it's actually affected the widely used RT-PCR test that I've already been telling you about. So now my talk is going full circle. I'm now linking the mutations we see in viruses and the new variants we're worried about to the PCR tests, and now explains why I told you about this PCR test. So if you remember, this thermo-TAC-path PCR test is looking for the presence of 3 viral genes here, green, yellow, and red. And the yellow one is for the spike protein. And it was found from September onwards in UK PCR test labs that the assay for the S protein was beginning to fail, and fail completely. And the other 2 tests were fine. So if we look at these graphs here, so this is what a normal positive test would look like. And you watch the yellow line now, that if you have the 6970 deletion, then this S gene would drop out of the assay. So this is the slang term used by the labs, S dropout, and you may have seen this in the media. And the reason that this S gene is not PCRing up anymore, it's not amplifying anymore, is because the 6 base deletion that takes away these 2 amino acids also takes away one of the primer probe binding sites. Right? These primers and probes can only bind to the viral sequence if the matching sequence is still present. If the matching sequence changes, if there's a significant mutation, you now take away the binding site for any one of those primers and probes. So if any one of those 3 doesn't bind anymore in the PCR will fail. And that's what's happening. So the genomic sequencing labs in the UK have sequenced thousands of samples where there was a positive test, but there was an S dropout result, and found that 99% of those were due to the presence of this B117 variant strain. So in the UK, this S dropout result in the PCR test is a good proxy assay to rapidly detect the spread of this new B117 strain. So this is really important for healthcare providers and for governments when thinking about how to balance lockdown measures, because genomic sequencing takes 2 to 3 weeks to go through the whole process and get results, and then it often takes longer to get enough results to inform policymakers that a new variant is on the rise. So whereas the PCR test only takes 1 or 2 days and is done right at the beginning of a patient presenting with an illness, and so you can find it very quickly that S dropout was only 1% of the population last month, but now it's 20%, now it's 30, 40, 50%. And that's exactly what's happened in the UK, is this S dropout has become a dominant feature of PCR tests, because this B117 strain has been spreading so quickly. And that has led the UK now to increase its lockdown measures. And unfortunately, we're in restrictions that are similar to March and April of 2020. And it's currently the start of January 2021. So I'll just end here with a few points, and hopefully now you've gained enough knowledge about the basic molecular biology to understand what a PCR test is, what mutations are, and why some mutations are more important than others. I just want to end to point out that there are these 2 new variant strains that are spreading very quickly in the UK and South Africa. It's known that the UK strain is present in many European countries. It's been found in the USA, it's been found in Australia. So unfortunately, it's already crossed borders, and a lot of scientists expect it to spread in other areas in the coming weeks and months. And the rapid spread of these two strains increases the challenge to governments and healthcare providers to keep infections at a manageable level where their healthcare systems can cope. And so we're going to have a challenging few months ahead until vaccines become widely used and we start to gain some immunity. There is no evidence so far that any of the variants that have been seen in any coronaviruses around the world would prevent the current vaccines from working as intended. So any of the vaccines that have already been approved, or any of the vaccines that are at an advanced stage of development, they should all work very well. It's important to point out that something like the spike protein is predicted to have 20 or so different epitopes on it, 20 or so different places where a human antibody could bind to it. Any mutants, it's likely only going to take away one or two of those sites. So a vaccine should still work very well. So there's nothing to be worried about in the long term. So future videos from us will look in detail at how the sequencing of coronavirus genomes is being used to track the spread and the relative risk of new coronavirus variants. And that will focus much more on the scientific data and the official reports coming out of mostly UK research. And so if you're really interested in what's been going on and what is informing government policy, then please do look out for those videos. Thanks for listening. Stay safe.